All right, let's resume from where we left from the first part of this lecture. So we talked about the different models of replication. We talked about the different step in DNA replication. So now what we're going to conclude this lecture with is talking about how do eukaryotic cells deal with progressive shortening of chromosome in some specific cells through the use of an enzyme called telomerase. And then we'll talk about the different DNA repair mechanisms and finally, the organization of the DNA in eukaryotes. So let's talk about telomerase first. One problem that eukaryotic organisms have is not a real problem, but it is a problem in some cells, is that because of the way DNA replication is done with a lagging strand that relies always on new primers, is that the chromosomes in eukaryotes would tend progressively to get shorter. If you look at the picture on the right side here, let me just select the pointer here. So you can see how this is how, imagine this being a eukaryotic chromosome. It will be replicated through different origin of replication, but imagine this one being just one in this case. So now you have that in the laggy strand, you have a DNA here. While this replication fork progresses towards this right side, we rely on more and more and more RNA primers. The problem you have is that when you get close to the end of this chromosome, the last primer doesn't always, RNA primer, doesn't always coincide with the end of the template strand. It might be placed sometimes a little bit further down compared. And so what happens in this case that when these DNA molecules, they will get replaced with DNA. When this gets replaced, so then when this DNA molecule becomes now the template for the next molecule, this will be copied, continues all the way to the end, but you would have created here a shortening of this chromosome, which progressively will get bigger and bigger over time. This is a normal process in somatic cells, right? The problem you have with this is that over time, here, imagine this is a chromosome, you have important genes here necessarily for the function of the cell, the survival and the function of the cells. Over time, since these chromosomes are getting shorter and shorter, at a certain point you would have that some genes will get lost. And therefore, these cells will get senescent and eventually die over time. This is perfect. That's a great mechanism to give the cells a certain amount of time of survival. That's perfect for somatic cells. For example, think about your skin cells. Your skin cells are exposed to radiation all the time. They get lots of mutation. Do you really want them to survive forever? No, you want them to die over time. And this mechanism indeed helps with their death. It gets them to die after some time. Once they have accumulated enough radiation, or even before they accumulated radiation, they die, they're done, they get replaced by new cells. However, this is a problem for some of those cells, such as your germ cells. Your germ cells are those cells that give origin to the next generation. Talking about sperm cells, egg cells. Do you want them to have a shorter DNA? No, because it means that with every generation you'll be losing DNA. So after X number of generation, you will lose some genes, right? You don't want that to happen. The same thing, you don't want this to happen in those cells, the stem cells, which are present throughout your body, everywhere, and also obviously in the embryo. These cells that give origin to the differentiated cell. For example, going back to the example of the skin, these cells that die in your skin, they shed all the time. Some people shed even more, they have dandruffs and so on. These cells that are shedding all the time, they have to be replaced. And they're replaced by cells that are at the very bottom of your epidermis. You have this cell, this stem cell, that do produce these new cells. But these stem cells, you don't want them to start losing DNA. Otherwise, they would die over time, right? So you want them to be able to keep their DNA intact so that when they produce these differentiated cells, these skin cells that then they shed, these skin cells will start with enough DNA to be functional, and then in those cells, the somatic cells, it can shorten up and die, but the stem cells, they have to keep this full DNA. How do they do that? They do that through the help of this enzyme called DNA polymerase. This enzyme, what it does, it prevents this shortening. How does it work? Well, first we have to talk about telomeres. At the end, both ends of each chromosome in eukaryotes, you have the sequence called telomeres. Telomeres are non-coding sequences, so they don't give origin to any protein. They're there just to keep the ends of the chromosome intact. And what they consist of is of hundreds of thousands of these repeats over and over. TTAGG, TTAGG, TTAGG. I'm not going to repeat it hundreds of thousands of times, but you get the point. You have this repeat present there over and over and over. So these are, if you want, disposable sequences. So in somatic cells, these sequences will get less and less. You get less and less of these repeats present with every single division. That's fine. They're non-coding sequences. You can lose them over time. At a certain point, though, once you run out of them, at this point, you start chewing through the genes. That's the problem. The cell will get senescence and then will die. But these are important sequences. Okay, so they are disposable sequences at the end of each chromosome that get progressively lost throughout each cell division, as you can see in this picture. Now, these sequences are also those sequences that telomerase act on. As the name says, telomerase acts on telomeres. How does it act? So basically, it keeps them, or it even expands them in germ and stem cells. This process we're talking about here is very active only or almost exclusively in germ cells and stem cells, and unfortunately in cancer cells too. That's how cancer cells that prevent their death, by reactivating telomeres. So this enzyme is highly expressed in these cells, not in somatic cells. And how does it do? So basically what it does, it keeps these telomeres from shortening, actually can even extend them. How does it work? Well, imagine this being your chromosome that has undergone DNA replication. Now you have that, unfortunately, this part here has not been copied because the RNA primer was just going up to here. Okay, so now telomerase can extend this sequence, but even the other strand by using its own RNA template. So this enzyme, telomerase, has within its active site this RNA template, which is, not surprisingly, is complementary to these repeats that make up the telomeres. So being complementary, these RNA strands can actually form a double helix with the extension of the chromosome here. By doing that, now, once this is done, the enzyme telomerase can actually extend this strand here. So start adding new nucleotide complementary to its own RNA strand, right? By doing that, it's basically extending the telomeres in the germ and stem cells. Now, this keeps going on and on and on. So it adds a few repeats, then the RNA primer is moved and so on. So it can really add more and more of these repeats, extending the telomeres. Once it is done, after this, so after it is done, now this gap on the other strand, you can imagine how basically extending a single strand DNA here. Once the RNA primer has been removed with the telomerase itself is going to get released, now this single strand DNA is then copied exactly like is done in DNA replication. You would have now room for another RNA primers to be added here, and then DNA polymerase 3 to act, extend the RNA primer. RNA polymerase 1 will go in, remove the primer eventually, or you can even lose it. Ligase works, it's gonna get all joined together. So by doing that, originally you had a sequence up to here. By doing that, this end of the chromosome actually has been not only preserved, but even extended in some situations. So by doing that, telomerase acting with its own RNA template, it will extend or preserve the end, the telomeres of these chromosomes in the germ and stem cells. Last thing we are going to talk, two things. DNA repair, first of all. DNA repair, as you can imagine, DNA is an extremely sensitive material. It's something you want to preserve extremely well. And unfortunately, or fortunately, it's good and bad at the same time. DNA tends to change over time. There are environmental factors. I mentioned before, radiation. The sun itself causes mutations on your DNA. But replication itself causes mutation. And by mutation, what we mean is the change of one or more nucleotides in the DNA sequence. This is may have different implications. We'll discuss them in the future lectures. But a change in a nucleotide may have no effect. But if it's in a coding sequence, so in one of those sequences that give origin to a protein, it may have devastating effect. It might change the protein, might change just one amino acid, but completely causing the proteins to completely lose the function. Not always. Sometimes it does, sometimes it does not. So these changes, these mutation, can have devastating effect. At the same time, they may also cause an advantage. Evolution acts on them. One change in one amino acid that might look devastating under different environmental circumstances can give an advantage to the organism that has it, allowing it to expand, selecting that population with that mutation for the future, causing evolution of that species. So there are these mutations that happen, and some of these mutations are introduced by the environment, as I mentioned, some of them by DNA replication. So how are they corrected? Organisms tend to avoid too much mutations, right? Now, there are two mechanisms or generic mechanisms. There are several mechanisms. One relies on the same enzyme that duplicates DNA, which is the DNA polymerase. It's called the DNA polymerase proofreading mechanism. Proofreading means it's reading what is just wrote, what it just did, and checks it and fixes it. And then there is a system of different enzymes and proteins called mismatch-based DNA repair mechanism that acts as well. So let's look at the DNA polymerase proofreading mechanism. Now, this was discovered by Kronberg and Bertlags, blah, blah, 1972. But basically how it works is while DNA polymerase, it's copying the template strand. It's adding one nucleotide at a time by adding it to the 3' hydroxy group. When it does that, ours is trying to match the template strand. Sometimes it makes a mistake. So it happens, all right? So 1 in 1,000 usually might make a mistake, introduce the wrong nucleotides. But then the DNA polymerase itself can sense a different instability. It's based on vibration, basically caused by the hydrogen bonds. So if you say you have a thymine, or in this example, you have a adenine here, and instead of adding a thymine here, DNA polymerase has added a cytosine. There's a mismatch there, right? It can sense. Now you have one that will form 3 hydrogen bonds with one that forms 2. You can feel the bulge in the 2 strands and the double helix and so on. So the DNA polymerase itself can sense that, okay? And when it senses that, it can actually back up by 1 nucleotide in this case and removes the wrong nucleotide I just added. This is referred to as the 3', 5' exonuclease activity of DNA polymerase 3 in this case. Backs up, removes that nucleotide, and replaces it with the correct one. Okay, so this is referred to as proofreading mechanism. Okay, that's what we're referring here. So the DNA polymerase itself can sense its own mistake, and by doing that, can reduce its error rate to 1 in a million. It's okay for prokaryotes. We're talking about genomes in the million of base pairs. So every cell division would introduce, let's say, few mutations. However, when you're talking about eukaryotes, this is not enough. 1 mutation for every million base pairs in a genome that consists of billions of nucleotides is too much. It means in every cell division, you would have thousands of mutations happening. Consider that the coding genes, the coding sequences are about 1 to 2%. Let's assume 1% of your genome. 1% with 1,000 of mutations there means that you're talking about tens of changes in each gene with each division. Too many, okay? You don't want that to happen. You might tolerate 1 every several divisions, but you don't want that to happen in every single cell division introducing 10 changes per gene. Too many. So how is this prevented? through a second mechanism, which is present only in eukaryotic cells, not in prokaryotes, which is referred to as the post-replication DNA repair mechanism, or repair mechanisms in general. So how does it work? Well, you have a series of proteins, enzymes, and other proteins involved here. That basically what they do, they're scanning the DNA, right, and sensing where there are changes to the expected structure of the double helix. So when you have a mismatch, base pair mismatch, like this one right here, thymine, guanine here, this will be sensed as some sort of bulge. There is instability there. So this complex here can sense that. And when they sense that, then they call for other proteins, other enzymes there that will actually remove several nucleotides around the area there. And then, so they would replace this gap here now. The DNA polymerase could replace it afterwards, and ligase could actually join these two strands together again, by basically replacing again the DNA to what it was done before. There are mechanisms through which this complex can sense what is the new and the old strand during DNA replication. This is a bit of a problem instead when it comes to DNA repair mechanism like this one for mutation introduced by the environment. You can see how in a situation like this, imagine that there is some UV light that changes bases, doesn't necessarily make regular bases, but change some that look like others and so on. Now, you wouldn't know which one is the original, which one is not. Which one is the one to replace, which one is not. So you could have that the DNA repair mechanism, even though they reintroduce a correct pair in terms of hydrogen bonds, they might still be introducing a new nucleotides where it was not supposed to be. So they could still have a mutation. You would have stable DNA, but not mutation. Now, the importance of this mechanism, as I said before, is that they reduce the mutations for each DNA replication to only a few mutations for each cycle, okay, which most of the time will end up in non-coding sequences. But you can see how when instead you have some mutation in any of the proteins involved in this process, you got a big problem. You start having lots of mutation happening in the people that have this type of mutations. And an example is in individuals affected by what is called xeroderm pigmentosum. This is a disease which is caused by genetic mutation. So sorry, not genetic, but by mutations in several of the proteins. It's not always the same protein. It can be one of the different proteins involved in this repair mechanism. So if you may have it on the one that sends the bulging, on this one that replace this or remove that. So there are different mutations that can lead to this type of disease. But you can see how the people that are affected by xeroderm pigmentosum, they're not able to repair the mutations. They're very ineffective at doing that. The mutation introduced by the environment, to a point that when they actually have to go outside, they have to be completely covered. They cannot be exposed to any mutagens like UV light that will cause, as you can see here, several mutation cause. Obviously, that's the name coming from pigmentosums, additional pigmentation of the skin reflecting mutation taking place in the skin cells. These individuals, obviously, they're very sensitive to mutation, and therefore they're very sensitive to different type of cancer, particularly skin cancer, when they're exposed to UV light. Final thing we are talking about here, DNA organization in eukaryotes. DNA itself in eukaryotes doesn't consist only of a sequence of nucleotides. Well, that's the DNA per se, but in reality, it's organized as a complex with other proteins. And the reason why it has to be complex with other proteins is exactly because of the large size of the DNA in eukaryotes. Just to give you an idea, your DNA is about 6 feet tall, 2 meters in each cell. Very long molecule. Can you really live like that? No. So it needs to be compacted. And the way it's compacted is compacted all the way to a fraction of a millimeter, 0.01 millimeter or 10 micrometers. Very, very tiny fractions, so they can fit inside the nucleus of your cell, is by having it wrapped around a series of proteins which are referred to as histons. Then there are not only histone proteins that what we'll focus on now, but there are also known histone proteins that help compacting this DNA to a point that it gets super compacted when mitosis happen or meiosis happens. We'll talk about that in the next few lectures. So, and when we talk about DNA complex with proteins, now we're talking about chromatin. Okay, we're referring to the complex itself. And that's what we really have in the nuclei. We have chromatin. DNA is always compacted with this protein, histone, and so on. So what are histones? Histones are positively charged proteins, okay, which they do interact. The reason why they need to be positively charged is that so that they can interact with the negative charge of the DNA coming from the phosphate group. By doing that, they can actually wrap around. There are 5 different types: H1, H2A, H2B, H3, and H4. And so they work in pairs. So, and what you have is that the DNA actually wrap around these 4 histone proteins or dimers, 8 of them altogether. So 2 H2, 2 H2B, 2 H3, and 2 H4 proteins compact together here. And the DNA wraps around this complex of 8 ultimately proteins, forming what is called a nucleosome. So this DNA wrap around here is referred to as the nucleosome. These already increase or decrease, if you want, the length of your DNA. In reality, it's not the end point. Now, these nucleosomes, first of all, are joined together by region, referred to as linkers here. They have a diameter of about 10 nanometers, and you have about 200 base pairs of DNA wrapped around. You can actually visualize them by electron microscopy. Like you can see here, this will be a thread of DNA wrapped around different histones here, forming the nucleosomes joined together by the linkers here. But now, this is not the end point. DNA will still be too long to fit inside your nuclei. And so these nucleosomes are actually joined together through the help of the other histone proteins. If you remember, there are 5 of them. These 4 are found in the nucleosome. And then you have the H1, which helps joining nucleosomes to go together. So the H1 histone protein brings them together, this nucleosome, causing them to further wrap around each other. So now you start having, because of the H1 protein here joining these nucleosomes together, you start having a structure consisting of 6 nucleosomes wrap around each other. And so this again extends the how compact your DNA can get into your nucleus. Now, these again can be visualized by electron microscopy. You can see how this structure here, these white dots here, will correspond to nucleosomes that get compacted together. These level of compactedness of DNA can change over time. I hope you understand how, obviously, if DNA is too compact, it will be inaccessible for those molecules, these enzymes that have to replicate the DNA, and they have to do the transcription as well, that they have to code ultimately for mRNA and then proteins. So DNA has to be accessible at times. So DNA tends to switch over time to one phase where it's loosely packed. In that case, we refer to as euchromatin, which will look like this, where you have a nucleosome but separated from each other, 2 times where it actually is very nicely compacted. In that case, we'll talk about heterochromatin. You can see this also by electron microscopy here. So this will be a picture of a nucleus. You can see the dense area here will correspond to heterochromatin, whereas the loose area here will correspond to euchromatin, where in this case, the DNA will be active, will be producing protein. In some cases, like in the X chrome in females, you have regions that are permanently basically inactivated, and they're referred to as the bar body. They're turned into heterochromatin and they stay like that for good. So they are completely inactivated. Otherwise, you would have an excessive dosage of some genes being expressed. And now there are several known histone proteins that are actually involved in the switching between euchromatin and heterochromatin, as well as the histone protein as well. It can be phosphorylated, methylated, acetylated, and so on, converting from one form to the other one. And you can see how, again, this is one of the many mechanisms through which DNA or the genes in the DNA can be turned on and off, can be expressed or not expressed. So finishing up here, summarizing both parts of this lecture, we talked about, first of all, an overview of DNA replication from a historical point of view. We discussed the three possible models through which DNA could be replicated, and we discussed the experiment by Meselson and Stahl, which ultimately made scientists understand that the true mechanism is a semi-conservative mechanism. Then we talk about the DNA replication, all the different steps. We talk about DNA polymers, how they work, in which direction, always 5' to 3', at least for the polymerase activity, not for the exonuclease activity. We talked about the different step, unwinding, elongation, termination, and all the factors, all the enzymes involved in those steps. And then we talked about briefly about the difference between prokaryotes and eukaryotes, particularly how you have different origin of replications in prokaryotes, whereas in eukaryotes you have multiple of one due to the different size. Then we talked about also how in eukaryotes telomerase takes care of the shortening of the chromosomes in the telomeres by basically keeping extending them in some specific cells, germ cells and stem cells, not in somatic cells. Then we talk about how excessive mutations are avoided by different DNA repair mechanisms. We went very lightly on that topic, but different mechanisms that prevent the excess of mutation in eukaryotic organisms. And then finally, we talk about how the DNA is organized in euchromatin and heterochromatin in eukaryotes through the help of histone proteins, the formation of nucleosomes, and so on.